So today we have a very nice treat for you and a lecture on biotechnology for humanitarian projects. And it's being presented by Dr. Namakanta Bhatt, who received his PhD in electrical engineering from Stanford University in 1996. He's currently a professor and the chair of the Center for Nanoscience and Engineering at the Indian Institute in Bangalore, India, the Indian Institute of Science in Bangalore, where he researches nano-electronic devices, electromechanical biosensors, and chemiresistive gas sensors. Dr. Bhatt is an IEEE fellow and has held numerous volunteer leadership positions within the IEEE Electron Devices Society, EDS, including his current roles as co-chair of the Nanotechnology Technical Committee, vice president of the Educational Activities Committee, and he's also a distinguished lecturer for the society. He is also the co-founder of the startup Sod Healthcare Private Limited, which has commercialized the first of its kind lab-on-palm device for diagnosing multiple chronic diseases. So without further ado, I would like to welcome Dr. Navakanta, but to deliver his talk on biotechnology for humanitarian projects.
Again, thank you very much, Fernando, for your kind introduction, and it's a great opportunity to give this webinar as a part of IEEE site initiative. Site, as all the audience understand, is a special interest group for humanitarian technology. And this particular webinar is on a general theme of biotechnology for humanitarian projects. Let me first acknowledge all my students and postdocs and staff who have contributed in some of the research that I'm going to present. There are a lot of funding agencies, both federal and private funding agencies, who have made this work possible. Fine. So this is the I hope you can see the screen. There is no issue, I suppose, with the screen sharing. Is that correct?
Yes, I can see your screen.
Okay, great. Yeah. So, what I want to sort of discuss in the next 45 to 50 minutes is mostly focused on diagnostic technology. While, biotechnology, as I will show you, is very vast, let's just focus on diagnostics. And in particular, we will talk about both communicable and non-communicable diseases and different techniques that are used in diagnostics, transduction technologies, the difference between doing tests in path lab versus, on the field, auto analyzers versus biosensors. Then I will focus on two specific examples. One, of course, is very relevant in the current context, which is to do with the communicable disease COVID-19, which is a pandemic now. I will briefly talk about two different techniques that are used to diagnose COVID-19. Then I will move on to non-communicable diseases, particularly anemia and malnutrition and diabetes and complications. I'll end my talk with some observation on future outlook. So as I said, biotechnology starts, uh, what we eat, food and agriculture. There is a lot of intervention of biotechnology there. Then, of course, diagnostics of diseases as well as therapeutics. Once you diagnose, then you will have the treatment, right? Again, biotechnology has played a crucial role in that. And also, a very important aspect is to do with epidemiology and surveillance. As a lot of you can appreciate, it has become extremely important in the context of COVID-19, right? However, what we are going to do in this talk, as I said, is only focus on diagnostics, right? That is what we are going to focus on. Okay. So when we talk of disease diagnostics, there can be two kinds of diseases, right? communicable diseases such as the flu that we get almost every season, and more severe kind of communicable diseases could be Ebola or the COVID-19 that we're all going through, right? Most of these are infections through microorganisms, right? Either virus, bacteria, and so on and so forth. The other class, which is of non-communicable diseases, they're not necessarily because of infections. Some of the non-communicable diseases are because of infections. You know, uh, However, quite a few non-communicable diseases are not related to infections. They are mostly often called lifestyle diseases, such as diabetes, for instance, cardiovascular disease, and also cancer, for instance, right? All these are non-communicable diseases. And in both these categories, diagnostics plays an extremely important role. The early diagnosis can make a big difference in terms of how we sort of deal with the disease and sort of eventually have this disease cured. Now, some very interesting data that I wanted to put across, if some of you are not already familiar, this is again thanks to what biotechnology has enabled over the years. Right? Until about maybe 5, 6 decades ago, communicable diseases were the major killers. But there has been a shift over the last few years, and the disease burden is really to do with non-communicable diseases today. In fact, NCDs, in short, are estimated to account for more than 70% of global deaths. Right? This is the data from the year 2018 from World Health Organization. You know, Out of this, of course, cardiovascular, cancer, diabetes, as I said, and complications of diabetes, which could also be cardiovascular disease, right? And communicable diseases are a much smaller fraction. That is something very, very interesting to note. And this is because, as I said, the advancement in biotechnology, which has happened in the last few decades, both in terms of diagnostics and therapeutics, right? This has really enabled this shift that has happened in terms of disease burden. So now, what have we witnessed in terms of biotechnology intervention in diagnostics, right? In my opinion, it's not just evolution. I mean, there has been a revolution in diagnostics, right? The way we used to diagnose, way back, let's say early 1900s, this is how a diagnostic lab would look, right? It would look like today's chemistry lab, right? Indeed, this is a place they would titrate blood and urine and would come up with the conditions, the disease conditions, right? But in the last few decades, thanks to what are known as immunoassays, we have very, very sophisticated equipment tool set that we use today. If you go to, either a LabCorp or a Quest Diagnostics, for instance, any of these places, you have these very sophisticated instruments. Most of them are actually based on what are called immunoassays. We will see what these immunoassays are in a minute. But this has been a very, very, very important transformation that has enabled, advancement in diagnostics, right? Now, immunoassay can also be called as the holy grail of diagnostics, right? Immunoassays, as when you say immunity, the first thing that comes to your mind is, antibodies that are produced in human bodies in response to microorganisms, right? Now, this interaction between antibody and any other foreign body, which oftentimes need not be foreign body, it could even be stuff inside the body, they're called antigens, right? So this interaction between body and antigen, which is called a binding event, is very, very, very specific. Right? Now, both antibodies and antigens are essentially proteins. As you know, proteins are building blocks of life. And the beauty between antibody antigen binding, as is shown in this picture here, it's like a lock and key arrangement, right? antibody, as is illustrated here, has this anchor or a tail. It's a Y-shaped structure, a protein structure that is, right? And at the ends, it has this capture sites, which is very, very unique, right? There are different kinds of antibodies for different kinds of antigens, and this particular antibody, for instance, can only capture antigen of kind A, right? So, antibodies, of course, are produced in human bodies as immune response, and that is why if a diagnostic test is based on antibodies, it's called immunoassay, right? That is how this name comes about, right? Now, specific antibodies are produced for different antigens. But what is very unique today in the context of biotechnology is that biotechnology has today enabled the production of antibodies and antigen in vitro. That is, we can produce them in large number, right, using various techniques outside the body and at will, sort of, right? And this is what is eventually going to be used in, diagnostic assays, right? So this is very important to keep in mind. Now, this uniqueness of antibody to a very specific antigen, as I said, lock and key arrangement is what gives the specificity so that you do not have any other interference, right? Now, there is something else required in addition to this antibody and antigen binding. And this is a classical diagnostic assay called ELISA, right? Which is also called enzyme linked immunosorbent assay, right? Of course, the fact that there is this word immuno, right? There is antibody and antigen interaction, right? That is given. However, just this binding of antibody and antigen does not necessarily produce a signal for detection. And that is why we say we need to label antibody or antigen, right? For instance, if you are trying to detect antigen, antibodies are your probes, you label that antibody with an enzyme, right? Now, these enzymes are also proteins, by the way. They're catalysts, really, catalysts for chemical reactions. And these enzymes, can be tagged or labeled to an antibody or an antigen, depending on which one is your probe. Okay? It becomes very clear if you're detecting a virus, then antibody which is specific to that virus is a probe. But if you're trying to detect an antibody itself, which is what we do in a serology test for COVID, then you need to have an antigen as a probe, you see? So these probes will be labeled or tagged with enzymes, and these enzymes in turn act on what is called a substrate. These are the languages used by chemists and biologists, right? To produce a measurable signal, right? For instance, a substrate we say may be chromogenic, which means when enzyme acts on this substrate, once this antibody antigen reaction happens and binding happens, then the color may change, which is what happens in the simplest diagnostic test that people use at home, like a pregnancy test, right? There's a change in color that you can sort of say that, okay, there is something that has happened, some marker has been detected. Or it could be fluorogenic, which means fluorescence is a detection technique, or it could be luminescent. No, it's just the emission of light. That could be a detection technique. These are different ways the enzymes, which are labels, act on the substrate. Right? The substrate is sort of converted into a product through an enzyme, and that is how you actually get the detection. For instance, here you have a virus to be detected. So I will use an antibody which is very specific to this virus. This antibody is tagged with an enzyme. Now this binding takes place. Now this enzyme then reacts with a substrate and in the process gives a detectable signal, right? The enzyme could be horseradish peroxidase, which is the most common enzyme that is used, or there could be other enzymes as well. There are also several kinds of substrate that can be, right? So this is one of the most fundamental way of detecting, the markers in disease diagnosis. Now we talked about this immunoassay and transformation. Going forward, what next is the question, right? I think what next is something that we have been talking about quite a bit in the last few years, is really to be able to have these variables which can be diagnosing your disease, your ailments on the move, right? So mobile platform, or home diagnostics, this is the direction that we are heading really, right? So now, in order to enable for us to move from pathology lab based big equipment to mobile diagnostics, what we need is something called biosensors, right? These biosensors are a little different from autoanalyzers. Autoanalyzers are the equipment that I showed you in the context of pathology lab, right? Now, a biosensor is essentially a simple device which has two components called receptor and transducer. Okay? And it takes a sample, which is essentially biological sample. It could be blood if you're doing a blood test. It could be urine. It could be saliva, for instance. It could be sweat. It could be tear. Or in the context of COVID-19, it could even be a nasal or oral swab, which is essentially trying to pick out this virus from your, no nostrils, for instance, inside your nostrils. So these are the different kinds of samples. Now, just because this is done outside on a mobile platform, you cannot compromise accuracy and efficiency, right? So obviously, you should have a capability to detect only a very specific molecule of interest, right? And then, of course, you need to also convert that into a signal, right, which is measurable, as we already talked about in the context of immunoassay, right? So now, if you look at this, there are different kinds of receptors. We already talked about enzymes and antibodies, which is what is used in the classical immunoassays, which is very, very common, extensively used in pathology labs. But you can also talk about different other molecules, some aptamers, specific ligands, some ions, chemical receptors, so on and so forth, right? In terms of transduction, in addition to simple color change that we talked about in the context of immunoassay, when I described that, you can have an electrical transduction, optical transduction. For example, fluorescence detection is an optical transduction. It could be acoustic, calorimetric, mechanical, and so on and so forth, right? So if we could build this, that will enable us in taking this diagnostics, biotechnology enabled diagnostic, to the next couple of decades on this mobile platform, right? So that is the direction that we want. So let me just, before I change the gear and talk about specific example, let me just then sum it up again. The elements of any disease diagnostics, in particular biosensor, you have a sample. The sample could be different, kind, blood, urine, and so on and so forth. And you have some receptor, antibody, enzyme, or different other elements as well. And then you have a transduction element, right? It could be a FET device, right? It could be a nanowire, carbon nanotube, nanoparticle. For example, gold nanoparticles are used in flow assay to provide color change. So this is a transduction. And then, of course, you need to have an electronic system, which is what we would eventually use to give out the signal, right? And these sensors can act over a very large length scale, starting from nucleic acid or DNA or RNA, which is of the order of nanometer, and going all the way to few micrometers or more, right, at the cell level, right? So the detection can sort of length scales also, right? So that is sort of a background on why this diagnostics is important, and different diseases, how the burden has shifted, importance of immunoassays, and also other receptors in terms of transduction, right? And eventually the biosensors enabling mobile platform in the years to come. Now let's look at two specific classes, communicable diseases let me just spend a few minutes on COVID-19 testing. As you know, COVID-19 is, I don't need to tell you much, we are all locked down at home and, maintaining social distancing and whatnot to ensure that the spread doesn't happen, right? So there are two kinds of tests that you must have read quite a bit in the literature. The beauty today, by the way, COVID-19 today, any literature is open source. Most of the journals have made it a policy that any paper that is being published is available for anybody to download, even if you're not subscribing to that journal, right? This is a beauty. So there's a lot of literature available for you to go and read. Now, there is antigen testing. This particular virus which causes this COVID-19 is, we'll come to that, coronavirus, which is a test called RT-PCR. You can also do what is called an antibody testing, using things like lateral flow assays. There are different techniques one can use. These are fundamentally two different things. We should remember that. Now, a lot of challenges in COVID-19 testing, right? Again, as I said, COVID-19 is the name of the disease. And the bug that is causing this disease is this virus, which is a coronavirus, belongs to this family of coronavirus. Particularly coronavirus 2, because there was another coronavirus which had caused, which was detected in 2003. This virus is an RNA virus, which means it doesn't have DNA. RNA is a building block. There is a specific RNA which codes the entire genome of this virus, right? Now, COVID-19 is a very severe acute respiratory syndrome. That is why it is so serious, right? People get admitted because of this respiratory problems, and eventually you may have to go on a intubation, or in the worst case on ventilator. This particular RNA virus has about 30,000 long base pairs, right? Both RNAs and DNAs are made of building blocks, nucleotides, right? And in this particular case, there are 30,000 unique base pairs which define this virus, right? Which is what is going to make this virus a little bit different from COV1, for example, right? There are a few differences in these nucleotides, and that gives its specific functions, right? Now, this is something very, very important to recognize. By the way, you can go to a lot of database today. They give a very nice three-dimensional structure. The reason why these viruses are called corona, they actually look like corona, right? This is how the structure looks. And there are a lot of proteins. There are some of them are called structural proteins. For instance, the spike protein is a structural protein. There are also proteins which do not take part in the structure of this, coronavirus, right? Now, of course, there is an RNA which sort of has the entire, code for, protein description of this virus, right? But the virus copies in a sample, when one is infected, can be as little as 15 viruses in milliliter of sample. So when you take a oral swab or a nasal swab, right? It's called either nasopharyngeal swab or oropharyngeal swab. You have only 15 virus copies in a milliliter. Very small sample you are taking anyway, right? Or in the worst case, you may have 150,000 copies. That's it. So this essentially means, in biology and chemistry, there is something called molarity. Okay. Molarity essentially means that when I say my molarity is 1 or 1 mole, it means that in 1 liter of that solution, I have 10 to the 23 copies. 10 to the 23 happens to be Avogadro's number, right? Now, as opposed to a molar or a millimolar, we are now talking about a femtomolar or ottomolar, 10 to the minus 15 or 10 to the minus 18. There is no detection technique which can detect this reliably. This is where the problem begins, right? There is other problem also which makes this whole thing very, very tricky. And that is the following, right? Now, the so-called viral load, initially you go through what is called an incubation period, where you don't even have symptoms, but you can still be spreading virus, and that is why it is so serious and scary, right? The incubation time can vary anywhere from 5 to 10 days, right? And then your symptoms start, right? And only then you start testing the virus, right? And eventually the viral load starts coming down. That is where your body starts producing antibodies. There are two kinds of antibodies that are produced. One of them is called IgM, which is shown here. They are first responder, if you will. And then little later, there is another class of antibody which is called IgG. All antibodies, as I said, are proteins. They're also called immunoglobulins, right? That is why this Ig, right? IgM and IgG are a little different, right? So this happens, and if these antibodies, along with the medicine, can control, then of course the viral load starts decreasing, and the patient begins to recover, right? Eventually, you can cure the disease, but this whole thing can take up to 4 to 5 weeks. You see, this is really very prolonged, right? So this is where we have the challenge, right? In order to detect this, then we need to resort to what is called an amplification, because as I told you, so few virus copies impossible to detect. Only if I can amplify. It's analogous to the amplification we do in electronics, right? You need to amplify the signal to see the signal, right? So this is where again biotechnology is so advanced, and it enables us to do this, right? Now, this is too small concentration. Let me just repeat that. To reliably detect the virus, number of virus copies should be amplified before the detection. And there is a technique to amplify DNA, not RNA, by the way. DNA amplification technique is available, which is routinely used, and it is called PCR. PCR stands for polymerase chain reaction. DNA, by the way, is a polymer eventually, right? Uh, you know, it- so this can be used by first converting the virus RNA into DNA. And this process is called reverse transcription, right? And that is why eventually it is called RT-PCR, RT-qPCR, because it can be quantitative. You can precisely tell how many copies of the virus are there in the person, right? So it's a quantitative detection, not qualitative detection, right? So what it does is that this polymerase chain reaction amplification brings the virus concentration nanomolar range, from atto to femto to nanomolar range, which can be now detected reliably, right? Using different techniques. One of the most important technique that is used is fluorescence, right? That is how we detect. And in order to come to this stage, we need about 20 to 40 cycles of amplification. I'll come to what I mean by cycle. Thermal cycling of ramping up and ramping down, right? So in a cartoon, I'm trying to illustrate this amplification process. This DNA is a double-stranded helical structure. I've simply sort of illustrated it as a simple parallel line structure, but it's actually a helical structure. There are four bits, A, C, T, G. It's essentially an abbreviation for these building blocks, right? So this is essentially a DNA, right? So what we need to do, take that virus, use this reverse transcription first. Whatever those 5 copies, 10 copies, 100 copies should get first converted to equivalent double-stranded DNA, right? Now we can resort to this PCR. What is So there is what is called a PCR mix. It's like cooking, really, right? So you have a solution. In this solution, you have what is called a template DNA. This is the template DNA, that is the DNA which needs to be amplified. You're starting with few copies of DNA. And there are what are called primers, right? Primers are essentially representing, see, I told you that there could be 30,000 bases, right? Primer could be just 10 or 15 base pair long, maybe 15 to 30 base pair long. So what is done is that you look at that whole genome sequence identify certain unique places in the genome sequence and create those single-stranded DNAs, I mean primers, right, which are 15 to 30 base pair long, right? And you also put these primers in the same soup. Now what you do, these primers, these are the primers here. This is a short primer and this is another short primer, right? So what I do first is that I dissect this double-stranded DNA into single-stranded counterpart. This is called denaturation. It is simply done by heating this soup to 95 degrees centigrade, right? And when you do that, it just dissociates. And when you cool it down to 50 degrees centigrade, this process is called annealing, right? When you do that, these two strands do not come and join each other. It's very difficult for these 30,000 base pairs to come together and join. But what happens is that this 15 to 30 short base pairs that you have uniquely defined can go and attach at the right place to this DNA, right? So now this becomes a starting point to build your whole DNA sequence. And that is done using what is called extension process, right? Which is done by again taking it to slightly higher temperature, which is about 70 degrees centigrade. What happens there is that you have an enzyme called Taq polymerase. It's essentially a kind of enzyme which facilitates chemical reaction. And you have these building blocks, which are essentially individual building blocks of the DNA. So it essentially takes these building blocks and stitches them at the right place. And that is how you essentially go from one DNA double strand segment to two of them after this entire cycle. This is called one cycle of PCR, right? So in each cycle, you are doubling the number. One becomes two, right? Now, how do you detect? Of course, there's the DNA. And the detection is typically done by adding another content, which is a dye, which is a fluorescent dye. It's actually called an intercalating dye. It has this property that it likes to come and sit in between the DNA nucleotides, right? So when you have very large number, only then this fluorescence light coming out starts becoming visible. That is why you have to go to certain number, n cycle. As I said, n is typically from 20 to 30 cycle. When you do that, the DNA copies increases by a factor 2 power n, right? If you do 20 cycles of this PCR, from 5 copies, you go to 5 times 2 into 2 to the power 20 copies, right? And now that is detectable, right? And that is what we detect in this uh, technique. And the reason why it takes, you may have heard that these tests are long, they take about a few hours, 4 hours, 5 hours, or whatever. This cooling and, heating all this soup that is about 20 to 40 microliter is actually taken in a plastic bottle, and it's not a good thermal conductor, right? This ramp rates are very slow and they take long time, right? And this is where again there is a lot of possibilities in terms of semiconductor technology to really miniaturize this PCR amplification going forward, right? So a typical RT qPCR equipment looks, and there is what is called a threshold cycle. You need to reach certain number of cycles. On x-axis you have cycles, on y-axis you have fluorescence intensity. Unless you reach certain number of cycles, if you have fewer copies to begin with, you need to go to larger number of cycles to get the threshold of the fluorescence. So you typically come across this term called CT, which is threshold cycle, right? And this is what you see. You get that only if you had that virus to begin with, you get the fluorescence, and you have detected the disease, right? This is, for instance, a typical test that is done on COVID patients. Days since onset of infection, this is a temporal that is done. This is a clinical study that is done. You see that different patients respond differently to nasal swabs and throat swabs, oral and nasal swabs. In some patient at on day 0, that is when you have symptoms, you can detect the virus maybe after 30 cycles. But in some patients, you cannot detect the cycle until you reach the 3rd day, right? But in order to do that, you have to go to 40 cycles, right? And this is peak viral load, and then as the time progresses, the viral load starts decreasing, right? Eventually, the virus disappears if you have cured the disease, right? So this is what is done in this technique, right? So the other test is what is called an antibody test, right? You should remember that this test is trying to detect antibodies in the blood which are generated in response to the virus infection. So it is not a technique to diagnose it early, right? This is very, very important. This is not useful for early diagnosis, right? But it is useful for epidemiology, right? You can still catch it fairly early when the person is still very infected, and at least start isolating them, quarantine them, right? And also, oftentimes, it is said that if you find these antibodies, then perhaps the person is already gone through this disease, and they have developed the immunity, and hence they can go back to work, right? You don't need to worry about containment and all that, right? So the way it is done again is that now, as I told you, there is this two-eyed antibody, IgM and IgG, right? They come at different times. The intent is really to detect these antibodies, right? So now you see the target is antibody, and hence to detect them, I need to have antigen in my immunoassay, right? If I were to do it through an immunoassay, right? Very important thing I want to highlight here. This is from one of the test kits which was used in Korea. Koreans are really credited in terms of doing early tests, right? Now, what I'm trying to show here is that these tests are not as accurate as RT-PCR tests, right? The typical sensitivities are of the order of 60%, for these antibody tests, right? Of course, if you wait for a long time, 3, 4 weeks, perhaps you have already cured the patient, then of course you will detect the antibody. Then of course you start approaching 90 to 100% kind of sensitivity, right? So it is not a good test for early detection, right? So the way it is done, it's typically like a pregnancy test kit, right? It's based on what is called a lateral flow assay, right? You have a lateral flow assay here, which means you put your blood or serum, right, onto a sample pad, right? And then you also put some reagent. Then this starts flowing laterally. Okay? And what you have done here is that you have actually put, as I told you, now we have the capability to artificially synthesize either antigens or antibodies, which are very specific to COVID and which are not contagious. They are not viruses, right? They're artificially synthesized, right? So now I'm using a COVID specific antigen, which is conjugated with gold, colloidal gold, which is what is going to give a specific color, right? And only if you have the right antibodies, then those antibodies will, bind to this antigen, right? And eventually you will have either the IgM or the IgG line shining up with some specific color, and that is how you are able to sort of detect this test, right? So this is what we use in antibody tests, right? So that is a very, very quick primer on using either RT-PCR or the lateral flow assay. These tests, by the way, are also used for various other ailments. I've used COVID-19 as an illustrative example here, right? Now let's move on to the other topic that I wanted to talk about. I'll just spend another 10 minutes to 15 minutes on electrochemical biosensor. Now these are different class of biosensor as opposed to the fluorescence based PCR or the immunoassay based lateral flow assay, right? This can be used again for a variety of diseases, mostly for illustrating it for non-communicable diseases. You can use it for anemia and malnutrition, for hemoglobin and serum albumin detection, diabetes complications, and so on and so forth, right? So how do these electrochemical biosensors work, right? Most of you have seen the glucometer. Glucometer has two components. One of them is a reader, right, which is an electronic meter, and the other component is this test strip, right? Now, it is the test strip which is the key, right, and which is what is going to do the magic, right, in terms of testing. So now, if you look at this test strip, what you see here is something called an electrochemical cell, right? This is essentially printed carbon based electrodes. These are long lines which are essentially going to take the signal from the electrochemical cell onto the reader that I have, right? And here you essentially have three of these electrodes called working electrode, reference electrode, and counter electrode. Working electrode is typically functionalized with a receptor. It could be an enzyme, for example. In glucose sensor, it is functionalized with an enzyme called glucose oxidase, which gives us very specific, detection capability for glucose. Right? You apply certain potential between reference and working electrode, and a current flows, which is proportional to the concentration of the glucose. Right, and that is how you do detection. Right. So you have an enzyme which is glucose oxidase, and this glucose oxidase, essentially, as I said, enzymes are catalysts, converts glucose into gluconic acid. All these reactions have electron transfer. So these electrons are eventually shuttled and eventually reach what is called a working electrode. And in the working electrode, these result in a current when you apply certain potential. And this results in a current which is proportional to the concentration of the glucose. And you essentially have a calibration curve which tells you that if you have particular value of current, that corresponds to certain value of glucose. Right? So this is how a simple electrochemical glucose sensor is. So, but what we see is that if you look at the electrochemical sensors, most of them are still glucose sensors, right? The glucose sensor market today, by the way, is more than 10 billion US dollars. It's huge, right? But other sensors are not as common, although there are publication increasing exponentially, right? And the reason for that is the fundamental building block in biosensor, which is receptor, right? You need to have that biorecognition macromolecule or receptor, which is robust and specific to a particular marker and also very stable in the field. Okay. So this is what we started looking at, and our thought process was that let's build not a glucometer but a lab on a palm, a mobile diagnostic platform, which can have multiple test strips for different ailments, right? So thereby you will be able to do multiple tests on a single device, right? So let's skip some of these here. So this is the kind of device that I will talk about, which is very useful for diabetes management and anemia and malnutrition, early diagnosis of kidney disease also, although I will not talk about it today, and also early diagnosis of liver disease, right? A very, very versatile platform at that, right? Now, this essentially works based on these test strips, but there are different test strips. There's a different test strip for a hemoglobin. There is a different test strip for a human serum albumin, different test strip for a A1C, and so on and so forth, right? So this is where you need to really put chemistry on this test strip. So this test strip is then inserted into the device, and then you have the right test done by the device, right? I'll just illustrate a couple of examples for hemoglobin. Why do we want to do hemoglobin sensing? Because is still a major burden worldwide. Okay, we are talking of biotechnology for humanitarian intervention, and the burden that anemia results in impaired development is just unimaginable, right? So if you can do an early diagnosis, easy diagnosis, that is the crux of this problem, right? So you see here, we are not trying to detect a bug, right? It's a protein which is already there in the body, but the protein is at very, very low level. It's below the normal range, and that is what we want to detect, right? Now, trying to detect is that this is a hemoglobin, and hemoglobin contains an heme center. That is why it's called hemoglobin. Heme center is surrounded by these globin chains. If you take out the globin chains, you can expose this heme center, right? So iron is a metal, as you know. So perhaps you can get an electron transfer between iron and your electrode. And indeed, you can do that. And this is how an electrochemistry of your hemoglobin would look. But the problem here is that if you do this electrochemistry, you will have a saturation of current at very, very low values of hemoglobin. Hemoglobin value of zero point 0.4, which is 20 times lower than clinically relevant lowest range. So it's of no use for clinically relevant hemoglobin detection. And this happens because at higher concentration of hemoglobin, you start blocking that electrode, and as a result of that, you don't get a linear current as a function of hemoglobin concentration due to this formation of what are called dimers and tetramers, right? So what we have been able to do is come up with an assay with the receptor, which is essentially to convert this hemoglobin into what is called hemichrome. There's a lot of chemistry involved here. We use a chemical called pyridine or imidazole, which belongs to a class of chemicals called N-heterocycle chemicals, right? And this can convert this heme into a structure called hemichrome. And then, if you do the detection of hemoglobin, you can actually span the entire clinically relevant range of hemoglobin. Remember that the clinically relevant range of hemoglobin for men it is from 12 to 14 gram per deciliter. From women, it may be perhaps from 8 to 12 gram per deciliter, right? So this test can span from even less than 5 all the way up to 25, right? So very large linearity over a very, very large range, right? So similarly, albumin biosensor, right? Again, we need to focus on the receptor, right? So if you look at albumin, unlike hemoglobin, albumin doesn't have any metal in it. It's not a metalloprotein. So it's very difficult to have electron transfer with albumin. Really, albumin can be called an insulator, right? Because you cannot really have an electron transfer. So that is why it is difficult to build an electrochemical albumin cells. What we have been able to do is to understand the properties of albumin in the body. Albumin is actually called a molecular taxi in the body. Albumin binds to a variety of substances, and we focused on binding of albumin to copper, right? Copper, because copper is electrochemically active, right? It's conductor, as you know, and albumin binds with the copper with the highest binding association constant, right? Which means all other interferers cannot really compete with copper, right? So I will not have any interference. That's the beauty. So what I've been able to do is build a sensor with this hypothesis that albumin binds to copper. While albumin is not electroactive, copper is redox active. If copper binds to albumin, that copper is not available for me for electrochemistry. And hence, if I measure the current from the copper, that should be an inverse function of the albumin concentration, right? And indeed, we did a lot of experiments and we verified that concentration does impact the current that we measure with an inverse relation as is illustrated, and eventually we are again able to sort of take it all the way to making a strip with the chemistry on the strip and validating this test strip with the biosensor that is essentially on the same platform. Now we talked about anemia and malnutrition. It is an equally worrisome disease today. Some statistics are scary across all countries. India is also called one of the diabetes capital of the world, but other countries are also, very, very severely affected by diabetes because it's called a lifestyle disease. While diabetes itself is, not a disease per se, it's a metabolic disorder, but it leads to all kinds of complications. The risk of stroke, risk of blindness, all increases because of diabetes, right? And that is why you need to detect diabetes and control diabetes, right? So the question was, what is available today for diabetes? Just the glucose measurement. It only tells you what is the glucose concentration at this juncture of measurement. That is not useful because glucose concentration in the body varies during the day. When you have some food and measure, it has very high concentration. Fasting glucose is low, and so on and so forth, right? So if you talk to the doctors, they tell you me glucose testing is not sufficient for me. I need other tests. For example, I need A1C test because A1C can give 3 month average glucose load on my body. And hence, doctor will know whether you are taking the medicine properly, whether you are doing the diet properly, and so on and so forth. And you can also extend this. There is another marker in the body which is called glycated albumin, which gives 2 weeks average glucose load on your body, right? So if you can do all this on a single device, then it gives a wealth of information to the doctor, right? So that is what we sort of set aside to do, right? So the way we have been able to do the HbA1c biosensor is sort of take the hemoglobin detection and marry it with what is called, boronic affinity principle, right? This, by the way, is used in pathology lab. Boronic acids such as phenyl boronic acid, aminophenyl boronic acid have affinity to sugars, right? So if your protein, like, what is glycated albumin or glycated hemoglobin? It's hemoglobin fraction which is complex with sugar, right? That is why it is called glycated or glycosylated hemoglobin. It's complex with sugar. So if you can trap this, you see, with this boronic acids, because they have affinity to this sugar-coated proteins, right? Then what you can do is that you can have two strips, right? Or in a single strip, two electrodes, one coated with just the hemoglobin chemistry, one coated with hemoglobin chemistry on top, coated with this trapping, which traps all the glycated fraction of the protein. Then what you could do is that you can measure total hemoglobin on one strip and the non-glycated hemoglobin because you have trapped the glycated fraction, and hence the difference can give you the glycated hemoglobin or A1C. A1C is essentially HbA1c is the glycated fraction of hemoglobin, right? So this is what again we have been able to do through a lot of optimization of the strips. And what is shown here is the pathology lab biorad equipment HbA1c measurement versus the point of care device HbA1c measurement shows excellent linearity with R square of 0.83, right? Okay, so that was about stress strips, and very quickly I will talk about this handheld reader. This handheld reader is a little more complex than glucometer. It's as if having multiple glucometer on a single device, you see, because we are trying to do many tests on a single device, you see, and that is why there are very interesting electron circuit design challenges, right? One can have very detailed discussion on that, but all I'm trying to tell you is that you need to have the right system option for your final solution, because your final solution should be at the right price point, if you're talking of the product, right? So you have to do a lot of optimization of your development, and eventually you build a device, you build a system, then you need to validate it. Medical devices have very stringent qualification processes, right? You need to take it through a lot of qualification processes. Your device may not qualify in the first pass, then you need to go back to drawing boards and try to improve the device build, and eventually you should be able to satisfy the required regulatory standards, right? And of course, the look and feel and user experience, all this is very important, right? So this is the device now which is able to do, as I said, 5 blood tests and 3 urine tests on a single device. And as I said, it's based on test strips. Making this test strip is also very, very challenging, right? I talked about science, but now you need to talk about manufacturing, right? So how do you manufacture them, right? Just like chip manufacturing, you need to manufacture them at very, very large scale. These are all printed, screen printed electrodes, right? And once these are manufactured at large scale, you need to convert them into sensor. And the way you do that is essentially you put a membrane on top of this, working electrode or an electrochemical cell, and the details of the membranes are extremely important, right? Because the membrane should be such that it absorb the liquid chemistry, but at the same time, it should let the blood or urine pass through, right? After mixing. So there are a lot of very, very interesting challenges when you talk of manufacturing, right? So a lot of optimization there. Eventually you would be, able to do all those optimization and uh, this the device. Just jumping a little fast. Again, as we were talking about the humanitarian impact, this device is now is deployed in several use cases. This has also been recognized by various fora. The biggest recognition is its deployment in resource challenge settings, right? Where you do not have labs, right? This is where such mobile labs will really be game changers, right? So, so what the user says is that the way they are able to do the medical interventions because of this device has really gone through a transformation, right? So this is a kind of, impact that the biotechnology, which of course is an interdisciplinary field. There is electronics involved, of course. That's where Electron Device Society is extremely important. Of course, there's biology, chemistry, and other aspects that are involved as well, right? So what is the future outlook? Everybody is talking of so-called digital health. That is the new buzz today. Digital health has a lot of aspects. Starting from health IT, electronics, medical records. Wearable and so on and so forth. Right. But we also need to come up with new robust assays, right? While IT and software and cloud, all those are very, very important. You need to come up with algorithm, but you're sort of leveraging existing know-how. But we also need to develop new know-how, new assays, new way of doing the tests, right? And then integrate with the rest of the platform, right? So there is a huge opportunity for so-called lab on chip technologies going forward. They have real transformation potential going forward, right? So with that, thank you very much. I think I've gone a little over time. I'm sorry about that. I was not looking at my time, but I hope it was very useful. Thank you very much.
Yeah, excellent. Yeah, so once again, I really want to thank you, Navakanta, for this, outstanding, a great lecture that I think, will be viewed many times on our webinar repository. So it really highlights, the contributions that the electronics and electron devices can make to the detection and to the current pandemic. And I hope everybody stays healthy and safe. And we thank you for your participation. And once again, thank you, Dr. Bhatt, for this great, outstanding talk. So with that, we will go ahead and close this webinar. Thank you all for attending.